We're going to take a look at the law of independent assortment. So this is another law that Mendel came up with. He figured out that the alleles of one gene will assort or are distributed into different gametes independently of another gene's alleles. And this is going to occur during meiosis in metaphase 1. So for example, if we look at two different genes, one gene with the coding for seed color and another gene coding for seed shape. So let's take a look at this in a picture. So the law of independent assortment occurs during meiosis, and this is because during metaphase 1 we get independent assortment of two different genes that are on the different chromosomes. So during metaphase 1 of meiosis 1, we could have our homologous pairs of chromosomes line up two different ways. So they could line up this way on the left, and you can see our two different genes. We've got our gene coding for the seed shape, the R gene, or the Y gene coding for the seed color. Or they could line up this other way on the right, and you can again see our two different genes for the seed shape and the seed color, so the R and the Y gene. And then when we separate our homologous pairs of chromosomes, after meiosis 1, here's the cells that we get. And then after we complete the second division in meiosis 2, here are the gametes that we end up with. So there's going to be 4 different possible gametes that we have. So these would be the eggs or the sperm, or if you're a plant, this would be the pollen. So our first possible gamete, we could end up with little r and little y together, and that would give us a gamete carrying the alleles for a wrinkled and green seed. And then we could also have this gamete with big R and big Y together. So this would give us a round and a yellow seed. And then we could also have this combination in a gamete. So we would have little r and big Y. So this would be wrinkled and yellow seed. And then we could also have this combination in a gamete, and this would be big R and little y, and so this would be a round and a green seed. So this is just showing the law of independent assortment, just showing you that the R and the Y genes will sort independently of each other. They're not connected to each other, so it's not like the big R and the big Y always have to go together and they always have to end up in the same gamete. So you can see that big R could end up with little y instead. Okay, so, and big Y can end up with little r instead. So there's different possibilities, different combinations, because the R and the Y genes, these two genes are sorting independently of each other. They can end up in different gametes. So that's all the law of independent assortment is talking about. Okay, and how Mendel figured out this law of independent assortment was he did what's called a dihybrid cross. So di meaning two and a hybrid is just a heterozygote. So he just took a pea plant that was heterozygote for two different genes. So what he did was he looked at two different traits. So he looked at seed shape and seed color. And so he just took two plants that were heterozygous for seed shape and seed color, and he crossed those as his two parents. And then what he saw in his crosses is that he ended up with this phenotype ratio. Remember, phenotype is what you look like. And he got this ratio with 9:3 to 3:1 with the offspring of this dihybrid cross. And that's how he figured out the law of independent assortment. Even though we're doing the law of independent assortment with this dihybrid cross and looking at two different genes, if you look at just the one gene and maybe you're looking at just the seed shape, you can still see that law of segregation where you see the 3-to-1 phenotype ratio if you're just looking at the round versus wrinkled seeds. So you're just looking at the round seeds versus the wrinkled seeds. And remember, the wrinkled seeds would be homozygous recessive, and then for our round seeds, they could be homozygous dominant or they could be heterozygous. And that phenotype ratio, that 3-to-1 ratio, we saw that with the law of segregation. So remember, if you're just looking at just one gene, you get that law of segregation where the R and little r will segregate or separate into separate gametes, and that is just showing you the law of segregation there. And also if you look at the yellow versus the green seeds, and you're looking at the Y versus little y, those two alleles, then you would also see that same 3-to-1 phenotype ratio indicating the law of segregation. Okay, and here's the dihybrid cross that Mendel did, and these are the parents that he was using. So he took these two parents that had round and yellow seeds. So you could see that they were dihybrids. So he looked at two different genes. So he looked at the round seeds and the color of the yellow seeds. And this is a much bigger Punnett square because we're looking at two different genes. So to get your possible gametes for these, what you would do is you would just look at both of these genes and you would say, well, big R could go with big Y, so there would be one gamete. And then you could say, well, big R could also go with little y, so there is another gamete. And then you could say, well, little r, we'll come over here just so we have more room, could go with big Y. And then little r could also go with little y. And there's your 4 possible gametes. And it would be the same with the other parent on the top here. And then when you cross these, you can see you have 2 big R's, 2 big Y's. And then if you cross this one with that one, you have 2 big R's and a big Y and a little y. And so you could do these crosses, and then if you count your offspring, you can see that you would have 9 that were yellow and round. And then you could see that there are 3 that are green and round. And then you can also see that there are 3 that are wrinkled in yellow. And then there's just 1 that is wrinkled in green. So that's where that 9 to 3 to 3 to 1 phenotype ratio comes from, so what they look like. And that's how Mendel figured out that how the law of independent assortment was working from that phenotype ratio. Okay, and then here's just a nice summary that talks about both the law of segregation and the law of independent assortment. So these are Mendel's 2 laws that he came up with. So how you tell the difference between these? The law of segregation is only talking about one gene or one trait that you're looking at. So maybe you're looking at just the seed color. Maybe you're looking at just this Y gene. Okay, all it's just saying is that these Y alleles, okay, from this one seed color gene are going to segregate or be put into separate gametes. Okay, they will not end up in the same gamete. You will never have a big Y and a little y together in a gamete. Okay, because that would mean the gamete would be diploid, and these have to be haploid. Okay, and the reason why the law of segregation happens is because of anaphase 1. Okay, because remember in anaphase 1, that is where you separate those homologous chromosomes. Okay, so big Y and little y, remember, end up in separate gametes. Okay, the law of independent assortment, how you tell this one, is you're looking at two different genes or two different traits. Okay, now you're looking at not only seed shape, whether they're round and wrinkled, but you're also looking at the seed color. Okay, and it's just saying that these two different genes are going to assort or be distributed into separate gametes independently of each other. Okay, so it's not like they're joined at the hip. It's not like big R always has to go with little y, like in this picture. It just says that, hey, they can line up this way during metaphase 1, or they can line up this way during metaphase 1. There's different ways that they can line up. So the law of independent assortment happens because of metaphase 1. Okay, so it's because of the way that these homologous pairs line up during metaphase 1. And so you can get these 4 different possible combinations of Y's and R's together, these alleles, when you have these two genes. Okay, but what about humans? Humans have 23 chromosomes in their gametes, don't they? So in humans, the number of possible gametes that we can produce is you take 2 and raise it to the 23rd power. Okay, and that'll give you the number of different possible combinations of chromosomes that we can have in our gametes. And that's going to be about 8.3 million different gametes that we can produce. Okay, so that is a crazy number. So that's just from one person. So you can see why your siblings are not going to be identical to you. So, because there's lots of different combinations for the eggs that can be produced by one woman, and then the man can produce lots of different eggs himself, or lots of different sperm himself. And then you've got random fertilization, and yeah, so lots of different combinations there.